Good morning, class. Today we are going to study the chapter life processes, and we are going to cover the topic digestion in small intestine. So if we are talking about small intestine, so complete digestion of carbohydrate, proteins and fat take place in small intestine. Actually, small intestine have various glands which secrete many digestive juices. So these glands are known as Brunner's glands and crypts of liver. So these secretes the digestive juices. The digestive juices are also known as succus entericus. Here, now these succus entericus contains various enzymes which help in digestion of carbohydrate, proteins and fats. Right? So let's study one by one. Here, maltase. Maltase is an enzyme which hydrolyzes maltose into glucose. It converts maltose into glucose. Got it? Next is sucrase. Sucrase hydrolyzes sucrose into monosaccharide. Hydrolysis means to say break down. It added the water and break down the substance. Right? So here, next is lactase. Lactase converts lactose into glucose and galactose. It breaks down lactose into glucose and galactose. The enzyme is lactase. Next is remaining fats. Remaining fats, actually here we have studied that emulsification of fat was done earlier also. So remaining fats which were not emulsified, so lipase you can say emulsify them into fatty acids and glycerol. Okay. Next, enterokinase. Enterokinase, it converts, you can say, trypsinogen. It is an inactive form of enzyme. It converts trypsinogen into trypsin, which is an active form of enzyme. Got it? So that it can function. Next is succus entericus. The digestive juices also contain few more enzymes, like you can say nuclease. It hydrolyzes the nucleic acid. Nucleotidase, it hydrolyzes the nucleotides, and nucleosidase, it hydrolyzes the nucleosides. So in this way, the whole process of digestion takes place in a small intestine and it is completed. Okay, I hope it is clear to you. And next we are going to study about the digestion in large intestine. We have studied the process of digestion in a small intestine. Now what happens next? The food goes to large intestine from small intestine. Actually, the whole digestion process was completed in small intestine, where food was digested, carbohydrates, protein and fats were digested and absorbed in the small intestine. Now, what happened to the food after that? After that, the food entered into large intestine. There actually whole absorption takes place in small intestine only. Now, the residue water is absorbed by the mucous lining of large intestine, and here the undigested residue, or you can say the undigested food, is changed into semi-solid feces. Now these semi-solid feces, you can say, move to rectum and they are temporarily stored there and they are ejected out of the anus, and this process is known as excretion. Now here it is finished. Now what happens to the absorbed food? We are going to study about the absorption of food. Right? Now absorption of food, so we can say that carbohydrates, fats, protein, which were converted into glucose, glycerol, fatty acids and amino acids. So what happens? These glucose, fructose, galactose, glycerol, fatty acid, vitamins diffuses through intestinal wall into the blood stream. They are absorbed into the blood. Okay, now they are transported to cells, various cells. Now glucose and fatty acid, they do not enter the blood, they enter into the lymph, which is present in lymph vessel. Okay. Now how this absorption take place? Actually, for efficient absorption, large intestine and small intestine, basically small intestine we are talking about, it is extremely long. It is extremely long and presents a large absorbing surface to the food. Its absorbing surface is further increased by presence of numerous minute finger-like projections called villi. Villi are the finger-like projection which increases the surface area. Suppose it might be cylindrical, so it do not have a large surface area. To increase this surface area, what happens? There are minute finger-like projections. These are present so that they can increase the surface area and absorption can take place faster. Okay, so now these are the villi. Let's study the diagram of the villi. So if we are talking about villi, it is a finger-like projection, single finger-like projection. It is villi. It contains blood vessels like arteriole and venous. Arteriole, artery, and venous is a vein. So here, vein are divided into arteriole and venous, and here in you can say between blood capillaries are present. Here, whatever the food is there, here it is absorbed and it is transported into the blood. Here, lactea. You can see in the middle, it is lactea. It is a lymph vessel in which lymph is present. You can say lymph. It absorbs fat and fat. It absorbs the fatty acids and fat. Right? They are transported into the lymph, and its single is called Villus, and here there are, you can say, microvilli present on each villi, which increases the surface area more. These are hair-like projections here. So due to increase in surface area, so the absorption process is faster. As I told you, each villus has lymph vessel for the absorption of fat and fatty acid. So this was the whole process of absorption. Now what happened next? After absorption, assimilation. Assimilation means utilization of the food we have absorbed. Assimilation, we can say here, now whatever the glucose was there, it is used in respiration to produce energy, right? And which is the excess glucose? The excess glucose is converted into glycogen and is stored in the liver and muscle cells. Okay, and whenever energy is required, further energy is required, so it is break down and is used. Next, amino acids. Amino acids are converted into protein. These proteins are used as enzymes for the constructions of various cell organelles, and in this way, proteins help in bodybuilding. So that's why we call protein as a bodybuilding material, right? Next, fatty acids. Fatty acids, you can see on reaching fatty acids and glycerol on reaching the cells gets converted into fats and they are stored.